afternoon, Dr. Devendra, sir. This is Jeevan from Reddy Women's College.
Yeah, good afternoon, ma'am.
Good afternoon, sir.
Yeah, so am I audible?
Yes, sir, you are audible, sir.
Okay.
In 2 minutes, we'll start the program, sir.
Yeah, no problem, ma'am.
As participants are joining, so just
No.
we'll wait for another 5 minutes and then we'll.
Okay.
Okay, sir. Thank you, sir. Sir, shall we start, sir?
Yeah, ma'am, we can start.
Okay, sir. Okay. Good afternoon, everyone. I welcome all the participants to the third day National Faculty Program on the role of DNA and molecular docking drug design. So today we'll be having a session by Dr. Devendra Kumar Daked, Assistant Professor, Naipur, Kolkata. Sir will be delivering a lecture on molecular docking and its applications in structure-based drug discovery. Now I invite Ms. Krishnaveni, Assistant Professor in Department of Chemistry, RBVR Women's College, to introduce today's guest speaker to the audience.
Thank you, ma'am, for giving this opportunity. It's my great honor to introduce Devendra Kumar Daket on behalf of Department of Chemistry, RBVR Women's College. Sir is presently working as an assistant professor in Naipur, Calcutta. Sir completed his B Pharm from Mohanlal Sukadia University, Udaipur, and M Pharm Pharmaceutical Chemistry, Bombay College of Pharmacy, and PhD Medicinal Chemistry, National Institute of Pharmaceutical Education and Research. He qualified GATE 2005, All India rank 24, and he qualified CSR NET in 2007. He's completed his PhD research project, Theoretical Studies of Conformational Tautomeric and Zwitterionic Preferences of some medicinally important molecules, and also completed DS Kotari postdoctoral research project, Discovery and Optimization of Novel Anti-Tubercular Agents targeting GIMU and structural analysis of Rhamnose pathway of Mycobacterium tuberculosis. And he completed postdoctoral fellow National Cancer Institute, Frederick MD USA during 2015 to 2016. Sir attended many conferences and workshops, ACS National Meeting, Exposition 2017, Washington, DC, USA. And he attended DST Sub Winter School on Modeling Chemical and Biological Activity in 2017, IIIT Hyderabad. And Sir has many research publications. So his publications in Journal of Chemistry, Informatics, JOC, and RSA Advanced Journal. And he has many skill activities, docking and pharmacophore mapping. Thank you, sir. Welcome you once again for FTP.
Yeah, thank you, ma'am, for the kind
opportunity.
introduction, and thank you for this, ma'am Jyoti ma'am, for contacting me, and my colleague Dr. Karnakar, who has contacted me regarding this program. So thank you, everyone. Is slides visible?
Yes, sir, it is visible.
Yeah, thank you, ma'am. So good afternoon, everyone. So today I'm going to present my talk on or discussion on molecular docking and its application in structure-based drug discovery in this FDP organized by RBVRR Women's College, Hyderabad. So before going to the docking, here is a general classification of the major techniques or methods which are being used in the CAD field, computer-aided drug designing. So based on the information, if we have regarding 3D structure of a macromolecular target, if we have, then we call it structure-based drug designing methods. And if we do not have any information about structure of a target protein or DNA, RNA, then we have to conduct the ligand-based drug designing methods, or we have to use them. So if we have the 3D structure, then we can do the molecular docking. If we do it at the using large number of the molecules, then we call it a virtual screen that is quite fast. And if we have the 3D structure, we can perform pharmacophore modeling using the 3D structure. And if we are interested to identify novel molecules from the scratch, then we can use the de novo drug designing methods, and later we can apply the molecular dynamics methods. And if we do not have any information about 3D structure of a target protein, then of course we have to use the ligand-based drug designing methods, and of which like QSR is quite common and popular. In addition to that, we can also perform ligand-based pharmacophore modelings and similarity searching and scaffold hopping as per our requirement. So in this classification today, we are going to discuss about the molecular docking. So molecular docking means, as we have understood, that it is the macromolecular target based technique. So we should have information about macromolecular target. And then this target may be like any receptor, enzyme, protein, any other DNA, RNA. and we should have some ligands. So our goal will be how this ligand is going to interact with the protein or this target, how it will interact, what would be the different intermolecular forces, what would be the conformation of this ligand in the active site. And we call this conformation POJ. This is the one of the major objective of the docking method is to determination of a POJ of a molecule. In addition, we have to determine what is the binding strength, what is the binding affinity of this POJ in the active site. So these are the two major things we have to perform, we have to determine in this method. So we have a protein, and protein must have the some site where we will explore the binding of the ligand. So suppose this is a one target molecule and we have the multiple ligands molecule, and based on their structures, these ligand will bind differently from each other. So here we see these are the three ligands A, B, C. So if we compare these ligands, so we can see here ligand A is perfectly fitting. There is a quite interactions overlap is there in the surface of the ligand as well as the protein. But if we see the ligand B, here we can see it is not a perfect overlap. And here are the two pose of the ligand C. So if we compare these two pose, we can say, okay, this C pose has got a better interaction compared to the pose C1. So this way we can differentiate between binding affinity and posage of the dissimilar ligand, whether we have to choose the better ligand or poor ligand. So this way we can differentiate between the poor and good ligands. And even if we want to compare the which is the best pose in our conformation in the active site, so we have to compare the multiple pose of the same ligand. Okay, so this way we can rank all the docked ligand in the active site of a given target using the different softwares. So there are two terms which are quite similar to each other, like binding mode and binding site. So binding site means this is the main site or active site. It may be catalytic site if it is an enzyme which is catalyzing the any reaction. So this is the place where the ligand is going to bind, and it is usually cavity or usually buried inside the protein. Sometimes it will be present at the interface of the two chain. And if by chance any ligand is known to bond with that protein, then we can easily identify, okay, this is the binding site in this protein, and one protein may have the multiple cavities. Out of those many cavities, only one is very important for our purpose. Others may be active and inactive based on the target. So suppose if we don't have any information, where is the binding site, where is the active site, there are several tools and servers are available. With the help of them, we can identify the probable druggable sites in a given target. So binding mode means this is the pose of a molecule or pose of a molecule in the active site. So this is dealing with overall the what is the geometry of a given ligand or docked ligand in the active site. The geometry means what is the location of molecule in the active site, what is the orientation of that molecule, and what is the conformation of that particular molecule in the active site. So this way we'll know location, orientation, conformation of a molecule in the active site. So here is just example I have shown. This is the one protein asparagine synthetase, and that catalyze that is a natural substrate for this enzyme. So this is one of the major cavity present in this enzyme, and here I have highlighted zoom in structure is there. Here we can see, okay, this is the asparagine is binding in the active site. So recently one survey is carried out about the nature of the active site. So more than 300 diverse protein were selected for this analysis. So they find out that active site usually may involve from the like 5 to 60 residue in the active site, but most of the protein they found that 20 was the very highest. 20 number of the 20 amino acids was quite common in the active site of those most diverse proteins. And if we see what is the nature of those different amino acids, as we can see, these belongs to the all major category out of those 20 amino acids, like non-polar, polar, charged, uncharged, and the aromatics. So binding site. So identification of binding site is very important, and we will see some of their important properties. So like if binding, if it is a enzyme, it should have a catalytic site. Some of the important residue, especially like acidic basic residue, because those are involved or in the catalyzing the or transformation or migration of the proton. So they should have some catalytic residue in the active site. If it is a protein, then it should have some important residue binding with them. It should trigger some action. Okay. So there are some other property regulatory mechanisms. So some of the enzymes have the allosteric site. So this kind of the enzymes have the two sites. One is the main site, which we call the active site, but the function of the active site is regulated by other site. If any ligand is binding here, it will lead to some conformation changes in the active site. And analysis indicated that these mostly binding site in these enzymes and protein are present in the major cleft of the protein or enzyme. And analysis also indicated that there is a usually safe complementary between the bound ligand and the active site. If we have the more complementary, chances of getting higher activity more compared with the ligands which has got a less complementary with the shape and size of a active site. Sometimes flexibility is also very important things in the proteins and enzymes. And if flexible loops are far from the binding site, active site, then there is no problem. But if any loop is there near to the active site, then it may be challenging. And there are several kinases are there which have the very dynamic loop and that present in the DFG in and DFG out conformer. So as you can see here, when there is no ligand, this is the one of the loop is present here. But whenever any substrate is bound and that substrate is interacting, then this loop move here and make a perfect cavity. Another important thing is that because these proteins enzymes have got their unique function, and that functions is determined by the conservation of some of the residue present in the active site. So if we compare the different targets of protein enzymes, we will see some conserved residue in the active site. Surface characteristics. In addition to the safe complementary, there are the electrostatic features are preserved in the interaction of a ligand with the protein. Means the negative residue of a protein will interact the positive residue of a ligand. So I have covered several slides here. Yeah. So the interaction between once ligand is bound to a protein or active site, and that interaction between, we can determine what is the strength of binding of particular ligand in the active site with the help of the some potentials or force field methods. Buffer zone. So as I said that if we have the more complementary between the ligand and the active site, then chances of getting better, more active ligand is more. Here are some of the examples which are the natural substrate or cofactors of their respective targets. So here we see, suppose this is the ATP molecule and it binds in these three different protein targets. And we can see this ATP has got a different conformers in the different protein, and we see different amount of the space on this between the surface of the ligand and the active site. Here we see quite empty space is there here too, and here we see in the bottom of this active site and this region, and here we have the large amount of the empty space. So this space we call the buffer zone, and that is present in the most of the between the substrate and the active sites of different proteins. Here is the FAD. Here we can see that if we compare these three structures, there is a huge amount of the space is available at the top of the ligand. But here, if we see in the second structure, it's almost perfectly fitting to the active site of this target or protein. And this is the example of one of the steroid molecule. And here we can see there is almost perfect fitting. But if we see here, we have the huge space here in the right hand side and the active side. So this is the buffer zone usually is present. But if we can use that, it can provide you better fitting and better activity to a molecule. So as we know that these target, usually if these are the proteins or enzymes are made of the 20 standard amino acids, and out of those 20 standard amino acids, residues which are present in the active site or allosteric site are quite conserved compared to the residues which are present in other part or surface of a protein. So here we see the ligand is bound and this is the active site, and we see the more darkest area here. So this is the most conserved area, and if we go away from this binding site toward the surface or far from the active site, these parts are quite varying and we do not see much conservation among the residues. So electrostatic potential I was talking about. So this is also very important, and nature has defined the how these natural substrate can have the opposite charge compared to their active site counterpart. So here we have the three example: adenosine monophosphate, heme, and estradiol. So these are the ballistic model for these different ligands, and here we can see this is these red colors indicating oxygens, and these nitrogens are in blue color. So if we see the electrostatic potential map from the active site, so here we can see this is the blue or red color, this indicating negative potential, but in the active site has got a positive potentials. Here we have the nitrogen rich area, but active site has got a negative potential to have a favorable interaction. This is the example of the heme center, which we know it has got a 4 pyrrole ring in center with the iron, and that keeping the one oxygen, and it can have the some carboxylic acid change. So complementary electrostatic potential is here, and here we have the 4 nitrogen in the center, so we have the opposite charges here. Similarly, in the case of the estradiols, at the terminals we have the 2 electronegative centers, and if we see the active site of this respective target, and we have the electropositive centers. So binding of any ligand to any target and also multiple or different types of the intermolecular interactions. Okay, so this is the protein and this is the ligand. When they are interacting, we are binding or interacting with the help of some of the non-covalent forces. If we have the more number of these forces, we can have the tighter affinity. If we have the less number of the intermolecular forces, we can have the loose binding, and by just conformation changes, that ligand can easily release. So here we can see there are 3 representation are shown. It has got only 3 interaction with protein B. Here we have the 4, and here we have the many. So we can say here we have the tight binding, more specificity of a ligand with particular target. Here we have the low specificity. By just the motion of the protein, these ligand may release, they may not produce any biological action. So here I have listed various possible molecular interactions that may be present between the ligand and the protein. So like ionic interaction, dipole-dipole interaction, and the dipole-dipole may be like between 2 dipoles, or interaction of any cation anion with the dipole, hydrogen bonding interaction, and this is one of the most common and easily visualized in the active site, and we can easily count how many hydrogen bonds are present. So it is present usually if hydrogen is present between 2 electronegative centers. If by chance electrostatic interaction and hydrogen bonding interactions are coming together, then we can have the salt bridge interaction between the ligand and protein. So here is one example where we can see there is a hydrogen at this nitrogen as well as a negative center. So it will make hydrogen bond as well as ionic interaction. So we will call it a salt bridge interaction. Hydrophobic interaction between the non-polar groups. Van der Waals forces, just because of the perturbation in the electron clouds, there is a generation of a temporary dipole moment, and that lead to the attraction between the opposite poles. Pi-pi interaction, if we have by chance two rings coming together, like aromatic rings coming together, they may interact, they may lead to the formation of a pi-pi bond, and there are different ways how these different pi ring can interact. Means like this is perfect sandwich model, this is sliced sandwich model. There may be T-type interaction between the two phenyl rings, because as we know that there are several aromatic residues are there, like phenylalanine, tyrosine, and tryptophan, and these three residues may interact with any aromatic system if it is present in the ligand. In addition, there is one variation of this. This is cation anion pi interaction. So any cation or anion can interact with this pi cloud. Like here we can see this is a benzene ring. This is the electron cloud or electron density. It may interact with the any anion. It may interact with any ligand which has got a positive center, right? Similar, this if we have any electron deficient ring, it may interact with the any anion. Halogen bonding. So if we have any halogens in the ligand, because halogens, we have the 4 halogens. Fluorine is the smallest halogen, and from chlorine, bromine, iodine, these are the 3 different or larger halogens, out of which iodine is the largest halogen. So because of polarization of the electron density, there is generation of some sigma hole on the tip of that particular atom, and that's electron deficient. Okay, suppose this is the halogen, and there is
Sir, sorry to
generation.
disturb, sir.
Yeah.
The slides are not moving, sir.
Okay, slides are not moving, so I can reshare it, so it will work. I hope.
Okay, sir. And sir, please increase your volume, sir, because we are able to hear, but little bit it is low, sir.
Okay, fine.
Okay, thank you, sir. Sorry for disturbing.
Yeah, no problem. So can you see now slides?
Yes, sir. Various molecular interactions.
Okay, now slides are moving.
Yes, sir. They are moving now.
Okay, thank you. So like we have the hydrogen bonding interaction between, like hydrogen is working as a electron deficient center, and this is the lone pair from the electronegative atom. It can interact easily with this electron deficient hydrogen. On similar concept, we have the halogen bonding, where this electron deficient halogen, especially chlorine, bromine, iodine, can interact with any of the electronegative center which has got a lone pairs, like especially like phosphorus, nitrogen, sulfur. On the same line covalent interactions, we usually do not see formation of a covalent bond between the ligand and protein. But today I will show you some of the examples where we can have such type of the interaction, and based on that we have the one type of the molecular docking methods. So overall, what can be the flow of molecular docking? So that is represented here. So in order to carry out molecular docking experiment, we should have the protein, we should have the ligand. So we have the proteins, we have the ligands. Then we have to prepare them. So we have to do 2 experiment, protein preparation independently from the ligand preparation. Then binding site definition, identification of a binding site. If you are lucky and you have got a protein which has got a co-crystallized ligand, then your job is quite easy because you know where the ligand can bind. Otherwise, a protein may have the multiple binding sites. In case you don't know where is the binding site, then you have to take help of some of the software servers. What are the different druggable site in this target? Then structural water, because these proteins are present in the physical conditions and they may have some special water molecule in the active site, and it is very difficult to identify which is the special water molecule which is conserved in the active site. But with the help of some of the tools, we can easily identify, okay, this is the important water molecule in the active site. And if you have sufficient literature, even when people determine the crystal structure, they usually mention about these conserved water molecules. Then we have, with the help of the molecular docking softwares, there are large number of the docking softwares are available. Some of the free, some of the commercial softwares. With the help of them, we can do this molecular docking experiment. Then we can do post docking analysis, how this software is predicting the binding mode of the different ligands. So first step is the ligand preparation, because this is the one of the important component of the molecular docking. So ligand preparation means it usually includes generation, optimization, and validation of the 3D structures. So if you have like handful of the molecules, or if you by chance have drawn those different molecules in the ChemDraw, so there is no problem the duplicates, right? You know what I have drawn, these are the serial numbers of those 20 molecules, or just I have to do docking of the 5 molecules, so we know there is no duplicates. But if you are doing molecular docking experiment of the thousands of molecule, millions molecules, then there is chance that those molecules may have some of the duplicate structures, and those duplicate structure may be in the term of like those structures are reported, some of the structures are reported with some cation, some with anion, some with some solvent. So we have to take care whether the database or your collection of the molecule which you are going for docking, whether it has got any duplicate structures. Ionization changes. If your molecule is very neutral, then it won't ionize in any condition, no matter what is the pH. And by chance, if your molecule has got a acidic basic center, then of course, based on the pH, it will ionize. And it may be possible, it may have the isolated charge center, means it may be present in the deuterionic form. And the deuterionic form will have the positive negative center, and that may affect the binding interaction with the target molecule. Protomer, if your molecule has got a acidic center only, or it has got only basic center, then it will go protonation, deprotonation, especially if it has got a basic centers like amines are there, pyridine is there, right? And amine, it may be like a linear and cyclic, primary, secondary, tertiary. So it will be going to be protonated. So we have to take care of that. Tautomer generation. So you may be aware like molecule may exist in the two different or multiple forms based on the migration of a hydrogen atom along with the reorganization of a single and double bond. So ketoenol is the quite popular form, but in addition to that, there are several variety of the tautomeric phenomena are reported in the literature in the organic molecules like azohydrazone is also there, which is quite common in the molecule which has got a azo bond, especially in the dyes. So we have to take care about the tautomers of a that particular molecule. Isomer generation. If your molecule has got some of the centers which are not clearly defined, right, then you have to include the both percentage. Like if your molecule has got one stereo center, but you don't know whether it was a R enantiomer, whether it was a S enantiomer, then you have to take both of them, because that database has not defined the stereochemistry of that particular center. Conformational sampling. Usually while we do docking, there are algorithms are there which do the conformational changes, but there are some of the softwares which are dependent on the pre-enumerated libraries of the conformers of a ligands. Like here I have given example Fred and Flock, which require the pre-generated libraries of the molecules. And if you have got a very large number of the molecules, of course you will not go for wasting your time, and because you have millions of billions of molecules, then you can't wait because you do not have much resource. So it is better to apply the some of the filters like drug-like filters, lead-like filters. Some of the toxicity related filters are there. By using them, at least we can reduce the number. So we whatever molecule we will have which are quite reliable. Energy minimization. So because when we are performing molecular docking, there is no change in the bond length between the atoms. So we should have the perfect bond length between the atoms, because overall energy depend on the force field parameters, right? If we take wrong geometrical parameter, then overall those things will be affected. So like as I mentioned here, tautomer and protomer differ in safe functional groups, surface and hydrogen bonding interaction, right? So whenever any ligand is binding to the active site, suppose it has got some of the heteroatoms, hydroxyl group amine, and it can participate in the formation of a hydrogen bonding interaction. Because of tautomerism or protomers, that hydrogen has migrated at different place. Now that particular atom may not be working as a hydrogen bond donor. Now it may be working as a hydrogen bond acceptor. So we have to be a little bit careful about these things because these are very important and crucial that may affect the binding behavior, binding mode of that particular ligand. Molecular structures. So source for these ligands may be like, it may come from the like ligand. One ligand at least may be present in the PDB, or if you have multiple proteins for that particular target, and then you can check whether there are multiple reference ligands are present for that particular target. You can draw those molecules in the ChemDraw, or if you have got it from some database, then it may be available sometime in the 3D coordinates or 2D coordinates or sometime 1D coordinates. Then you have to convert those all the structure, whether it's 1D or 2D in the 3D form, right? So whatever the structures we are generating by converting these 1D or 2D structure to 3D, those structures should be realistic. Those bond angle, bond length should be in the equilibrium distances and equilibrium bond angle and radial angles. After conversion with some softwares, then we have to optimize those starting structures to get a proper geometry of them. So if we go from the 1D toward the 3D, then complexity structure is increasing, but we have more information because in reality, this is the 3D structure which is interacting with the target molecule. Small molecule databases. So if you are interested to identify some new lead molecule for your particular work or target molecules, so as you may be aware already, there are several small molecule chemical databases are there, and which has got a huge number of the molecules, and that may have like thousands molecules, may have the millions molecules, and they may have the billions molecules. So already some of the database already categorizes different libraries and based on the target specific, based on their property like lead-like molecule, drug-like molecule, non-toxic molecule, something based on some filters. And if you are interested to dock multiple databases, then you have to be careful because there is a chances of the duplicates, because one database also has got a molecule A and database other database also has got a molecule A. So there is always chances of having a molecular duplicates if we are using multiple databases. So we have to be careful about if we are interested to do docking of the multiple databases. Each database has got some unique features. That's why that particular person or that company or group or university has generated that particular databases. So rather than doing docking for using only single database, it is better to use a multiple database. So we will have some structural diversity for the ligands. So therefore it is advised to use the multiple libraries while carrying out this molecular docking experiments. In addition to that, there are several natural products are there, and there are so many drugs are there which are not following the Lipinski rule of 5. Whenever this Lipinski rule of 5 or rule of 5 was given in the 1997, there were hardly, I think, 3,000 drugs molecule. Out of the 3,000 drugs, more than 90% drugs following that rule. But after the now 20 years has gone, more than 20 years has gone, now several drugs has approved which are not following Lipinski rule of 5. So we have to be careful, like if we have to do docking of the several molecules, then we can apply them. And if we have the handful of the molecules, then we should not bother about this rule. So recently people have proposed what is the chemical space of the molecules. So they have proposed that there are 10 to the power 64 possible molecules in this universe. So we have several library which has already have the collection of the synthetic molecules. We have several databases which have the collected the molecules different natural sources. There are several other biologicals are there which especially like antibodies or peptide like molecules. Out of these several possible chemicals, we have very small volume of the druggable molecules which follow like which has got a drug like property and do not have any toxicophore substructure. In addition to that, now people has started to enumerate all those possible chemicals, though those chemicals may not be available in the self, right? So based on the possible reactions, like we know there are several reactions are there in organic chemistry. So based on their chemistry, people have generated libraries of the billions molecules. So those we call theoretical molecules or virtual molecules or virtual libraries. That's why I say like there is a possibility of having 10 to the power 64 molecules. So there was one recent review somewhere, I think in Nature, they have collected some of the information regarding the different databases. So if we see this red color space, so Merck company has got the biggest their own proprietary databases where they have 10 to the power 20 molecules. Nobody knows what is the structure because this is their private property. Then AstraZeneca has got a huge number, 10 to the power 17, and similar online there, Eli Lilly and other company has got a big numbers. So these are the big companies because they are doing research from several years and they have different units. These are some of the others commercial databases like Enamine real space. They has got 10 to the power 9 molecules. Enamine real database. And several others are there. I don't know why this zinc has come in this category because zinc is freely available, developed by the University of California. So if we see this graph, big numbers are there. And if we see number of the approved drug, including FDA drugs and of drug approved only Russia, approved in the China and Europe, total number comes somewhere in 10,000, right? That is small dot. This is very strange. So here is a list of some of the other publicly freely available chemical databases. GDB. So one group in the Germany, they have recently released a very big database which has got a 10 to the power 11 molecules. This is the NCI SAVI, which has got a 10 to the power 8 molecules. So this is the my postdoc supervisor group that has developed this database. So what is the specific feature of this database? That they have just used the 64 coupling reactions like Suzuki coupling, gamma Suzuki coupling like reactions, and based on the most popular building blocks of the enamine and the sigma aldrich, and using some of the basic empirical rules, whether this, if we have such functional group, then this reaction is possible. If we have electron withdrawing, then this reaction may not be possible. Using those rules, they have generated this library NCI synthetically archive of virtual inventory, something name they have given. Recently they published in the Nature. Then we have the PubChem. This is also coming from the NIH. That's one of the premier institute. So this has got, I think, more than 100 million molecule now. On the same line, we have several other databases which has got a good number like enamine, NCI Open, Campbell, SENEX, Cambridge Structural Database. This is the not a public database, but this database has got a chemical structures which are mostly solved by X-ray crystallography. Campbell has got around 22 million structures, which are manually curated structures, and every structure has got some activity. NCI Open, this was the first database in this world. People have started collecting the structures of a molecule. So this is the place where I have worked during my postdoc. So we were there ligand preparation, and then we have gone to the chemical databases, and while preparing the ligand we have to be quite cautious. So here you see this is the one structure is there, and if we use it, you see here it has got some counter structure. Okay, some fragment is there. It may be any solvent counter anion, counter anion. So while preparing the molecule, we have to remove this counter structure. So next is the example where we see that there is no defined stereochemistry, right? So we have to generate the stereocenter across this bond. Here, this is the one tautomer, but because this molecule has got a possibility of the tautomerism, so we have to generate the tautomers. Here are other examples. Yeah, I think I have changed the ligand. So here in the last example, here we are generating the geometrical isomer. We don't know whether it's a cis or trans, so we have generated them correctly. And here we don't know what is the stereocenter, so we have to define the both possible, whether it may be R or S. So we have to define the both structure because we don't know what is the starting stereocenter. And here we have the possibility of the tautomer in this pyrazole ring. So of course, then we have to generate the possible tautomers across this ring. And here is the very special case. This is the very simple structure which has got a 3-member ring system and one ring attached to this nitrogen center. But this structure may present in the 2 isomeric structures because of isotrope isomerism. So in one case, this phenyl or hydroxyl group or phenolic group will be behind the this screen, and another possibility that it may be the in front of the screen. So we have to be careful such thing. Recently, one paper is published in the GMC. They found out that these are 2 different isomers which cannot be interconverted even by heating for the several hours. So if we have the handful of the molecules, so at least we should be careful about the several things. Another thing is metalloligands or metallodrugs. So far we have seen the most of the ligands which are based on the carbon chemistry, right? So those carbons may have like sp hybridization, sp2 hybridization, sp3 hybridization, and we can have the linear, tetragonal, planar, and tetrahedral geometry in the ligands. But we know long back that cisplatin is one of the most popular drug molecule for the cancer treatment, and that drug molecule has got a platin metal. So because these different heavy metals, especially from the D block, they have the quite varying oxidation state. So we can have the multiple type of the ligands. We can also fine tune different type of those ligands. We can have the heterogeneous ligands over this metallic center. So compared to these specific three geometry, these different metal organics or metal compounds can have the different variety of the structures, different geometries. So this way, people are nowadays trying to fine tune the selectivity of these metallodrugs. And these allow several other possible geometries in addition to what we have here seen, like pyramidal, bipyramidal, octahedral. And if we have the more coordination number, then we can also see more complex geometry across this ligand. So what we are doing, we have the target protein and we are seeing how this ligand is going to interact with the target. Metal is not present in the protein, but it is present in the ligand. So if your metallodrugs has got heavy metals, especially from the D orbital, D category, so then you have to be careful because those things cannot be handled by normal molecular mechanics method, normal force fields. So we have to go use of the quantum chemical methods, which are quite expensive, but whatever the geometrical parameter we will be getting, whatever the charge we are getting, that is more close to their things we can get from the experiments. So here I have listed the several popular molecules, some of the already approved drugs, some of the are the already in the clinical trials, like these are the different platinum based molecules approved, and these are different molecules which are currently in the clinical trials. Here are the some other molecules which are based on the other metals, like here I think it's arsenic somewhere, antimony, bismuth, silver, iron, like. So because if we prepare these ligands using simple molecular mechanism method, you won't get the proper distance, means especially this between the platinum oxygen, platinum nitrogen. If we use the quantum mechanical method, so DFT method, we will get the desired distance and other parameters. So now next part, now we have prepared our ligands. Now next part is the protein preparation. So first we have to identify what is our target, and we should have the 3D structure of that particular target. So we already know that there is a protein data bank is there that has got a 1,90,000 structure approximately. So if you are lucky enough, then you may get the structure of your target there. And by chance, if you do not have, there are several others way like homology modeling is there by which you can generate the 3D structure of your target. In case if you are not interested to generate the 3D structure of your target, now recently last year in the July, AlphaFold submitted more than 35,000 structure in last July. But recently, I think 10 days before, they have released again another site which has got a, I think 2 million structures. I think that is the maximum number that is present in the Uniport. So you may be already knowing the Uniport has got a sequence of different proteins. So they have solved the structure for for all those sequences available in the Unicode. So those structures are generated with the help of the deep learning, machine learning algorithms. So those are quite reliable. You can check even based on the basic stereochemical parameters of structures. So in this line, first thing is the structural integrity. So if you are getting structure from the protein data bank, so it may not be perfect. If you are lucky, then okay, everything is resolved. There is no missing residue. So we have to check whether any missing residue is there, missing segment or loop is there. And if it is there, we have to take care that with the help of the software or similar structures, we have to build those missing residues or loops if those are present in the active site or near to the active site. So next is the protonation and the tautomeric state of ionizable residues. So as we know that there are 20 amino acids, out of which there are 4 amino acids which come under category of the acidic and basic residue, like aspartate, glutamate. These are acidic residue. Arginine and lysine, these are the basic residues. So we can easily tell, okay, what will be the protonation state of this residue based on the pH. Very simple, no problem. But histidine is another base, but it is a weak base. So it is quite challenging to define the protonation state of the histidine. So because histidine has got a, I think, a pyrrole ring and it means two nitrogens, so that proton, that hydrogen may be on any of the nitrogen. And based on the micro environment, it may be protonated on the any of the nitrogen. So it is quite challenging to determine the protonation of the histidine. If it is reported in the literature, very good. Otherwise, with the help of the softwares, if it is present in the special inductive site, we have to be careful. So here, as we can see, this is the histidine residue, and like here, one nitrogen close to the side chain and one is the away. So this hydrogen over this nitrogen may be here, one possibility is, or one tautomer is there. Or this, whether this hydrogen is present to this nitrogen, then we can have this tautomer. Or this is present in the protonated form, then we will have the hydrogen on the both of the nitrogen. So out of these three possibilities, which tautomer is present? Because if this is present, your ligand is somewhere, then it may not or may interact. So this is the problem. So we have to be careful, otherwise our results will be affected. Next is the orientation of the ASN and JLN residue. So these are the two residues which has got a amide group or amide group in the side chain. So whenever we perform the X-ray crystallography, so because the amide group has got it, oxygen and nitrogen, two heteroatoms, so that it is very difficult to differentiate between the electron density for these two residue, because in the periodic table these two elements are coming together, right? So quality of data what we are getting from the X-ray crystallography for these two residues is not of a good quality, so there is chances that it may have the two possible conformers. We can flip oxygen to the nitrogen, nitrogen to the oxygen. So we can have the two possible for each amide bond for these two residues. So we have to check which orientation can provide some hydrogen bonding with the nearby residue. So it will finish the hydrogen bonding network, and that will be playing some role in the stability of a protein. And next is hydrogen atom addition. So as we know that the whatever the structure we are getting from the X-ray crystallography, we do not have the information about the hydrogens. Just we have the information from the heavy atoms. So we have to be careful while adding hydrogens. Then we have to optimize the structure, correct bond and bond orders. So by optimizing the structure, we have to correct the different bond angle, bond distance between the different atomic pairs, because sometimes loops are quite flexible, so we do not have the clear Cartesian coordinates, so bond length may be varying for different, like phenylalanine. So one case phenylalanine have the perfect distance between carbon that we have for aromatic bond. In another case, it may not be possible because we are not able to determine the exact coordinates of that structure. So we have to correct. There are several artifacts are there in the structure. Active site water molecules. So these water molecules may be present anywhere, and because these targets have the different multiple cavities, so those are filled with the water, and we don't know which water molecule is real or artifact based on the crystallographic data. Real means which may be conjured from or between among different proteins and may play important role in the stability of that protein or in the binding of any ligand with the protein. So it is quite difficult to judge which one is important, or it can be easily displaced or can be removed. So in general, rule of thumb is that if that particular water molecule is not able to interact with any nearby water molecule, or it is not able to make interaction with any ligand, if it is present in the active site, or it is not able to make any hydrogen bond interaction with the protein, then we can reject that water molecule. And if we have sufficient literature for that particular target, then we can easily check whether which water molecule is conserved in that particular protein target. In addition to that, we know that PDB file protein, that file has got a huge amount of the information about each atom and residue. So based on the B factor, we can determine whether this water molecule is tightly bound in the active site. So if we have several water molecules and by suddenly few water molecules, especially in the active site, has got a very low value of the B factor, we can say this is very important water molecule. We can keep it for our docking experiment. Set charge and atom types for the any metal atom. In the case of the ligand preparation, we have seen metal center was present in the drug molecule, or the molecules we are interested to design may have the metal molecule. Another possibility that there are possibilities that protein may have the metal center, and we call those proteins like metalloproteins or metal enzymes, like cytochromes are very important enzymes which are associated for the metabolism of the several drug molecules and other exogenous material. Similarly, there are several other proteins enzymes are there which has got a heavy metals like zinc, iron, cobalt, which are very important for their functioning or structural integrity. So if your target has got a metal, then it is quite challenging to handle such targets. There are several reasons. Some of them I have listed here, like multiple possible oxidation state. So because these belongs to the heavy metals, so which may have the quite variable oxidation state. Based on that, we can have the variable coordination or number of the ligands which may attach to this metal center. Sometimes there is a exchange of the ligands based on the change in the oxidation state. So ligand may add, may release. So this is the quite equilibrium between addition, deletion, or removal of a ligand. Main problem is the force field parameters for the geometry and flexity, because those minimization whatever we perform which are based on the force field methods, where we are using the force field, and those force fields are based on some Hooke's law. Usually Hooke's law related potentials are there which has got a bond length, angle, some constant parameter which are specific to the atom pairs. So because coordination number has changed, number of the ligands present on this metal has changed. So everything is changing. Therefore, we do not have many parameters, especially for the metals present in the active sites. So I think for zinc, people have determined the parameters. Others I am not aware, but it is quite challenging to have the good quality of the parameters. And because of these ligands are coming, going, so based on electronic features are also changing, which are in dynamic mode. So now we have prepared our ligand, we have prepared our protein. Now with the help of the software, we have to do our docking experiment. So we have to find out where is the cavity. So if that protein has got a co-crystallized ligand, we can easily identify, okay, this is our active site where we have to perform the molecular docking experiment. Then it is very easy task because we know the binding site. In case your protein of interest does not have the any bound ligand, then it is quite challenging where to dock. You can do the blind docking, means whatever the different sites are available, you do the docking to each site. Or with the help of the softwares, you determine which are the more druggable sites. So use the multiple software servers so that if all giving that, okay, this is the site one, is the druggable site, it has got a more druggable score. Then there is problem how I will validate my docking in the case where we do not have the bound ligand. Then you go to the literature and check if something is reported for that particular target, and for those inhibitors, if you can get the good pose, good score, and you can compare with the inactive molecules or less active molecules, then you can justify yourself, okay, whatever the experiment I am doing, it is quite reliable, because we do not have the bound crystal structure of a ligand. So once we have carried out the docking experiment with the help of the docking softwares, and then we have to value while doing, before going the docking experiment for the other molecules, we have to validate our the docking experiment whether the software is able to reproduce the already known conformer, already known geometry of a ligand in the active site. So for that purpose, we can extract the ligand from the protein, we should modify. So we can minimize the structure, optimize the structure, and we have to perform the docking experiment to the same protein, which we have already prepared. Then we have to compare this protein bound conformer with the docked conformer. And then we have to compute the RMSD. So here we have the three poses. So for these two poses, like orange and red, you see perfectly aligned. This phenyl ring, this indole ring, and this side chain is perfectly aligned. So we have this RMSD somewhere 1 angstrom. We have the additional pose in the blue color, and we see this phenyl ring is not able to align with this, and other part also going somewhere here, this indole ring, and it's supposed to be here. Therefore we have the very high value. So if we are able to get the low RMSD, so we can trust the software because it is able to reproduce the already known conformer of a bound ligand. If your software is not able to reproduce that pose, then we can't trust the software, because we know the facts. This is the non-conformer for particular ligand, and the software is not able to reproduce that, so we can't trust the software. So then what we can do, we can try to
Sir, we are unable to hear you, sir. Voice break. Sir, can you please rejoin, sir? Sir, you are in mute. Dear participants, please wait. Sir will be joining.
आप थोड़ी करा सकते हैं।
Dear participants, there is some network issue. Sir will join within 2 minutes. Your voice is not audible.
Hello. Yeah,
Yes, sir.
there was I think some network issues. So I joined by using my mobile. So I'm back.
Okay, sir. Okay.
So please allow me share my screen. There is some option is popping up. Host disabled the participant screen sharing something.
Sir, just a minute, sir.
Yeah.
Yes, sir, you can present now.
Okay. So whether these slides are visible now?
Yes, sir, it is visible.
Okay, sorry for this interruption. So we were, yes, I believe at the docking revalidation. So we have to validate our what target we are doing this experiment. Means if our software is able to validate, so we can trust the software because it is able to reproduce the original conformer reported in the protein data bank. So in case we are facing some problem to reproduce that conformers, then we have to fine tune the different available options or parameters with that software to get the desired results. So this is the RMSD. So as we have seen that this red and orange are perfectly aligned, and if we see this blue conformer and it is quite away here from the original this phenyl ring, so the distance is there. So this phenyl ring is not able to overlay with this red color phenyl ring. That's a crystal bound structure. And if we see this side chain, and here we can see there is a huge distance is there. So that's why we are getting huge RMSD for this blue color pose. So we have to be careful about the getting the right RMSD or to get a right pose similar to the structure bound pose. So there are several examples in the literature and where people were not able to get the or not were not able to reproduce the protein bound structure. So this is one example reported some review where from I've taken this picture. So with the help of gold, they have tried to reproduce this gray color is indicating this is the crystal structure bound pose or structure. And with the gold, they have tried docking of this peptide like structure. This is the structure 2D structure. So they were able to reproduce it. Though it was quite big structure, has got several rotable bonds. And this was another structure with like nitrophenol like peptides, and here they quite close to the X-ray structure bound pose or structure. But in this case, that the gold software was failed to reproduce the protein bound structure for particular fatty acid. So if you see this fatty acid, it has got many carbons, and at the end it has got a COOH group. And what that gold software did, it has flipped the orientation. Here is the original conformer where this is COOH group, but gold has proposed this is the best solution where this is the COOH group and tail is here, and tail is here, head is here. So it was failed for this target and for this molecule. On a similar line, there was a popular target in the HIV like integrase. So for that, there was only one structure sold in the 1997. So people were not able to reproduce the binding mode of that particular inhibitor. And that part people reported in the several structure and several papers reported in the JMC. So now visual inspection. So now we have done our docking experiment, and now post docking we have to check poses we are getting in the active site. So different people have their different strategies. Some rely on the score, some rely on the different things. So we can check like, is there any steric issues in the bound ligand and the protein? And in addition, we can also check electrostatic and hydrophobic complementary interactions. And next is the presence of hydrogen bonding interaction between ligand and the protein, and these interaction can be easily visualized. So if there is a hydrogen bond, and if it is present in the hydrophobic environments, then this can contribute significantly in the binding affinity, because this type of the interaction is not associated with the dissolution penalty. If this hydrogen bond is present somewhere like solvent exposed areas, then in that case those hydrogen bond donor receptor are highly populated with the solvent, and to make a hydrogen bond then we have to desolvate these hydrogen bond donor receptor, then that's a huge penalty we have to pay in order to make a hydrogen bond. So in by chance, if we have the possibility of making hydrogen bond and that hydrogen bond donor receptor present in hydrophobic environment, this is very good for the particular target. In addition to that, we have to check like electrostatic interaction with the metal cofactor. So whenever any metal enzyme is there and it has got a metal, there is chance that that metal will interact with the your doctor ligand. So we have to check that interaction. And in case if we have any conserved water molecule in the active site, so we have to check whether that water molecule is able to make any hydrogen bonding interaction with this dog ligand. In addition, there was a recent survey people have done with the computational chemist, which has got a less than 5 year of experience, or which has got a 5 to 10 year, or which people which have the more than 10 year of experience doing the such experiments like molecular docking or other experiments. They analyze what and how they analyze their docking experiments. So if you see this long bars are there, and if you see in this x-axis docking score, so people have given the less weightage to the docking score rather than they giving the preferences to several other things. Whether analogy to the crystal means when they are doing experiment, their ligand or their method, their docking software is able to revalidate or is able to reproduce the same conformer. They are checking complementary interaction. If for particular target, if a specific interaction is reported in the literature, then they are checking that whether the docked ligand is able to make or not. Ligand strength, electrostatic interactions, hydrophobic interactions. Some people are also checking strength of the interactions. They can compute by what is the strength or contribution of each interaction energy, number of the interactions, location where it is, several other like water mediated interaction, entropic contribution. So if anything is reported in the literature for particular target, so we should be aware before performing that experiment, so we can check whether the target is able to involve in that particular interaction. So we know that what is the source of the 3D structure for the protein. So usually whatever the structures we have in the protein data bank, usually these are coming from the three methods: X-ray, NMR, and cryo-EM. So mostly the structure are from X-ray followed by the NMR. Then now last 10 years people have started using the cryo-EM. That's a quite good method. With the help of that we can get the structure of very big or macromolecules. So this is the case we can do the X-ray crystallography if we can get the single crystal of the particular protein. Okay, then we can add the ligand, so we will get some idea about what. But we have what the problem? We are not able to get the positions of the hydrogens using X-ray crystallography. So what is this NMR spectroscopy? So this is the case we can use this method if we are not able to get the structure using the X-ray crystallography methods, because like membrane bound receptor we cannot crystallize them, then we can use NMR method for them. And there is no crystallization requirement, there is no lattice forces, and it is quite close to the physiological condition. Even you can reproduce by giving like a pH, ionic strength, temperature, that will quite close to the physiological condition. And it is quite less time consuming compared to the X-ray crystallography experiments. And usually we can resolve the position of the hydrogens quite easily compared to the X-ray crystallography. So there are some of the important parameters we have to look in the proteins, like resolution. And whenever we have the multiple entries for our target, we will see, okay, which one is the best structure for us, right? So here is a just a representation for the at different 5, 6 representation of some backbone along with some phenyl ring for particular protein that was solved at the different resolution level. So if that structure was solved at the 5 angstrom, then we can see we have some electron density for the backbone, some electron density, but what was there, we don't know. We do not have clear electron density. If we are 3 angstrom, we have some electron density that was initially missing between here. And if we have some resolution at the 10 angstrom, so we can get some clear picture compared to here, right? So in this case, we see, okay, there is a connection is there. Here, okay, there is no connection. There is no connection. And we see some clear electron density. Here there was just spherical structure. Now we have clear electron density across this phenyl ring, this side chain. Here we are not able to get the electron density for this side chain. And so we are getting clear picture as we are going the higher resolution, right? So if we can have that structure at this level, we can see that we are able to get some atomic resolution. We know, okay, some spheres for each individual atoms, not for all, but for most of the atoms, we are getting atomic type resolution. At 1 angstrom, we have the clear atomic resolution for each atom present in the structure. And even if we go further down here, we have the clear electron density for each element present in the structure. So this is the advantage. If we can have the structure with better resolution, we can trust our structure, protein structure, than our experiment. So resolution. So if we can get structure in this range, somewhere 1 or less than 1, we call it atomic resolution. And we have somewhere like 1 to 2, we call it a high resolution structure. And 2 to somewhere 2.5, we can say it's a medium resolution structure. And if you go beyond 3, it's a poor resolution structure, and we can't trust the results. So we have to be quite careful while choosing our protein. And this is the occupancy. So as we know that proteins are in dynamic nature, they are not static. So there is a huge motions, movement of side chain, loops, several things are happening. So even in the crystal, there is no clear sometime occupancy for each atom or functional group. So if we have the one occupancy, it means we know what is the exact location of the particular atom or functional group. Okay, but if we are getting multiple occupancy for particular functional group or amino acid, then it means we are not able to determine correct location, either that it is quite movement, so it has got a two different possibilities in the 3D structure. So here we can see this is amide. So this is one of the amide containing amino acid. So it has got a 2 occupancy like 0.57, 0.43. So this may be possible, this may be possible. If we compare these two, okay, we can consider this because we have the high preference for this location compared to the this location. And this is the another example where this phenylalanine has got it equal occupancy. This may be present in this area, or this phenyl ring may be present in this area. So it may affect your protein preparation. So we have to check, be quite careful which conformation we have to choose, or we can prepare the two proteins, one with this conformer, another with this conformer. Then we can check how the things are going, which conformer is allowing to reproduce your protein bound structure. So this is very, very important. So here are some other examples I collected. Just I think last month someone has published very good review regarding the side chain flexibility and protein flexibility. So here are some of the examples. As you can see, this is the arginine basic residue. So it has got a two different occupancy like 0.45 and 0.55. And this is another acidic residue. So like sometimes we get a protein structures from the protein data bank, and we have the homomeric structure. We have the multiple chains like A, B, C, D. So in one chain, you see this arginine is coming in this way. In another chain, like in B chain here, it is going this way. So as you can see, the protein is same, and different chains has got a different orientation conformer of those side chain of amino acids. This is the case of the lysozyme C. And this is the example where two different labs has crystallized this protein, lysozyme protein C, and from this species, Gallus. So they have got a two different geometry for this residue. One lab has got a conformer this way, and other member is able to get this confirmation for this side chain of this amide containing residue. They are similar other residue which also from different lab has got a different results. So we have to be quite careful. And if those residues are present, especially in the active site, then we have to check each carefully, which we have to consider. So another important factor is the protein, it is temperature factor, B factor. So because whatever the electron density we are getting using X-ray crystallography, it is not same for each atom. So because those atom functional group residues which are present on surface, we will get the clear electron density, better electron density compared to the residue which are present inside the protein. So if we are getting low B value, it means there is a tight packing. And if we are getting high B value, it means there is a motion in that particular area. So we have to watchful on that. So yeah, next is the docking classification. So far what we have seen, like docking of a small organic molecule to a target protein. So because protein can interact with another protein, can signal different things in several pathways are there which are based on the interactions of the protein. So we can have the protein docking. So we are just based on the surface interaction, we can determine how a protein can interact with other proteins. So this is very difficult task, right? Because one protein has got some residue, other protein has got some residue, and how to determine where they can interact, because they has got a big surface area. These are not few residue as we have seen in the case of the ligand, right? On the same line, we can have the protein ligand docking we have seen. Yeah. So type of the docking, so based on the flexibility, right? Because protein it is in the dynamic nature. So loops, side chains are moving, and when ligand is binding, it is also influenced by the forces of the active site. It can adopt the different conformer what we have seen in the computer. So based on the how we are treating flexibility, means for organic molecules, so simple like we can say protein or simple organic molecules, we can classify this way like rigid docking. So somewhere in the 1980, this concept of the docking was initiated, and then the people started the use of the concept like lock and key like concept. Okay, so in that both counterparts are kept as a rigid. We have the rigid protein, we have the rigid ligand. Then we are checking how those are interacting. And then because that has got several problems, we may not be able to get the desired results. Then flexible docking. So in that case, we have to keep the both things, target as well as the ligand, flexible. Next, the flexible ligand and rigid target docking. So in that case, ligand is kept flexible and our target is target molecule is kept rigid and the ligand is flexible. So this is thing. So next, the ensemble docking. So in that case, if by chance if we have the multiple structures available in the protein data bank, then we can use that information, because whatever single protein we are using, it has got a one particular conformation. but proteins are in dynamic nature, so they may have some other possible conformers, other different variation of the side chains in the active site. So if we are using multiple protein together, then that may give the better desired result. So we call it ensemble, means we are using collection of the some proteins to do our experiments. So based on their complexity, what we are doing, like if we have the both protein is rigid and ligand is rigid, then it is quite easy to perform, but quality of data may not be good. Okay, so another possibility that protein is rigid, but at least we are keeping the ligand flexible. So I think most of the software what we are mostly based on this concept. We usually keep the protein rigid and we try to keep our ligand flexible. So here we have the less complexity, but if we start keeping our protein flexible, then it will add more complexity to our experiment. Along with this, yeah, in this case both counterpart are flexible, so it is quite complex, it is quite time consuming. And if we keep this protein is flexible, again rigid, we may not get the desired conformer, because of protein we are not allowing whatever the conformer we are providing, it will bind in that. So like ensemble docking we have seen. So if protein data bank has got a multiple structure for your target, so what we can do, we can align all those together and we can do the docking experiment in those all. If you do not have any software which can perform the ensemble docking, and like some softwares already allowed to do the ensemble docking, like Gold, where you can load all the different proteins and it will allow to do the ensemble docking. So this is good idea to consider, or this is the one way how you include the protein flexibility in your experiment. It is better to have if you have the few ligands to dock, and if you have many ligands to dock, then it may be quite time consuming. So in case you are still interested in the performing the ensemble docking and you do not have the other proteins, right, just you have got only single protein structure, or if you do not have the another protein, then you have to generate the some other structure. So other way is that we can generate the multiple structure using the molecular dynamics experiment. So performing molecular dynamic experiment, we can get the different trajectory, we can cluster them, and then we can align and we can do the molecular docking experiment. So this is another way if you do not have the multiple copies of your structure in the protein data bank. So this is the new way we can do ensemble docking. In addition to that, it is quite close to this united protein system. If you have the multiple proteins, right, we can do ensemble docking. But similar to that, this is the system where we can take the best feature from the each protein, right? So here we have the three proteins, like and it has got three important regions, like part 1, 2, 3 belongs to the each protein. So this is conformer A, conformer B, and C. So if you dock, okay, you are doing docking of this ligand, and you see this ligand is not able to dock in these two proteins in yellow as well as this. So in this color, I don't know what to say. So you see there is a bump is there, the steric class is there, right? And even if you dock in this cyan color protein, it is fitting in this area, but it is not able to dock in this area because there is some conformation changes in this region. So what you can do, you can make a one perfect structure choosing the best features from the each conformer or from each protein, and then we call it a united protein concept. So then you will use that, okay, I can use in this area yellow color or this other color feature. Here I can use this cyan color feature. In this region, there is no problem. Even you can go up to the maximum space because you know this protein can adapt the any region from here to here. So this way we can do mixing, matching of the best feature of the active site. So we know that protein small docking was there, protein docking was there, and then in addition that we can have a protein peptide docking. So because here one is the protein, another is the short peptide, which may have like 5 to 50 amino acids. So there are different way how we can do the docking experiment for those peptides. So template based docking. So in that case, we have protein structure, and that protein structure has got already some bound ligand, that is a peptide. So we can do some modification in the side chains of those available residues in that peptide, and we can energy minimize. We can check the things how they are going. That's we call the template based docking. Otherwise, we can go for local docking and global docking. So for each, we have some of the list of the software by which we can perform. For that, we should have the information regarding binding site. Then our experiment may be quite easy. Otherwise, it may take long time to decide. So next is the nucleic acid. So this is the another important area. Now people are trying to already have identified some targets in the nucleic acid area. So we know there are two nucleic acids are DNA and RNA, and the DNA is associated with the cell division and responsible for the regulation of the several things, and RNA is associated with the transcription and translation. So if we can identify those targets, especially in DNA, RNA, if we can target them, then they know go behind this like protein inhibition, something. So there are some of the common mechanisms how those different ligand can bind with these target, which are based on DNA and RNA, like intercalation and cross-linking and strand cleavage. So as of now, there are, I think, more than 500 RNA ligand complex as well as more than 700 DNA ligand complex in the protein data bank. So docking in the nucleic acid not quite easy, as we have seen in the case of protein. So here I have listed some of the important things people are nowadays exploring, like DNA duplex, DNA quadruplex, where we have the 4 chain of the nucleotides, non-coding RNA, and this messenger RNA. So people now trying to identify these things to particular disease. And if we compare the DNA versus RNA, RNA is more flexible, has got a several loops. So usually DNA have got a duplex or quadruplex structure, but RNA do not have that, and it has got a several loops. If you can identify that particular loop which is druggable, then it is quite easy to perform the task. So we cannot use all the software whatever available for the protein docking, because things in the nucleic acids are different. Like here we have highly polarized region because of presence of the phosphates. Similarly, we can have the several metal ions which are responsible for the integrity of these nucleic acids. Yeah. So this I will end this docking of this DNA, and one more topic I will cover, and we have left with a few minutes. So this is the covalent docking. So far whatever type docking we have seen, it is based on the non-covalent interactions, which are like hydrogen bonding interactions, Hartree pi, and others, halogen bonding, hydrogen bonding. But covalent bond means we are going to make a sharing of electrons of a ligand with the protein. So only few specific functional group can react, like nucleophile and electrophiles. So there are some already possible reactions are there which can be possible. So if those features are present in your ligand and the active site of that particular target has got some of the important nucleophilic residue like cysteine, serine, tyrosine, then there is a possibility that ligand can bind by covalent bond formation. So here you see in this list, I have taken this list from the Schrodinger website, like cysteine is everywhere, and what can be the participating electrophilic centers, like here two nitrile groups are attached to this double bond. Here we have the alpha halo amides, and these are activated Michael acceptors, nitro group, and thiocyanide and ketonic group. So these all can participate in the covalent bonding, and this is the way how we can represent in the smarts. And this is the smarts for this nucleophilic residue, and this is the smarts for the ligand. So if you are interested to filter out those ligands which has got only this center, you can search using this in the PubChem, and you will get the only those structures which has got this structure. So then you can use those structure for carry out the covalent docking. So there are some other examples. You can easily visit the Schrödinger website. Here you will get examples. You can skip this slide. Here I have listed some of the most popular electrophiles which can participate in the covalent docking, like ketones, and if you have any strained ring like epoxide, aziridine ring. So here any nucleophile like cysteine can easily attack here and may lead to the covalent bond formation. Even if we have any sulfide group in the ligand, even it can participate in the disulfide bond formation. And if we have any halo alpha halo ketonic group, even that can participate in the nucleophilic substitution by SN1 or SN2. And there may be like nucleophilic addition, like if we have the ketonic group, like nitrile group can also participate in the nucleophilic addition across the nitrile group. So these are some of the common things recently one review people have reported, which are quite common in the covalent inhibitors. So these are some of the softwares which can be used for to carry out the covalent docking. Some are the, I think, commercial, some are freely available. I think Autodock is freely available. We can skip this. Last year, people have reported one structure for protease enzyme of the SARS-CoV-2, and they come to know that this is the one of the flavonoid molecule, and it was found that this is binding with that SARS-CoV-2 target by some covalent bond, and activity was somewhere in the some micromolar range. So how this ligand is able to bind? Okay, there is no clear, if you see the structure of that flavonoid, there is no clear any the structure what we have seen just 2 slides before, but because of tautomerism, this structure can provide the one hidden fragment that can participate in the covalent bond formation with that protein. So here we have the possibility of the 1,9 shift hydrogen migration that lead to the tautomerism, leading to generation of this quinone type structure, right? Then now what we have here, alpha beta unsaturated ketone Michael acceptor. Then that this any cysteine residue will attack here, will lead to the formation of the bond and removal of hydrogen, leading to the formation of this. So this is the proposed mechanism how this ligand may be binding. We don't know, but based on tautomerism, we can explain how that particular ligand may be binding to that target. And this re-tautomerize, and it will come to this normal structure where this is the aromatic ring. So docking we have seen, but what is the central part or central things in this method? This is the search algorithm and the scoring functions. These are the two things which are working together to provide the best approach along with the best binding affinity. There are several search algorithms are there by which software try to generate multiple structures in the active site, and those different structures are being evaluated with the help of the scoring functions. The scoring functions, we are giving some ranking, some numbering, we are giving, okay, this is the best geometry in this active site which provides the lowest energy structure for the particular target. So these two things are working together, and we are able to do our experiment successfully, so that such algorithms are helping in the generation of a pose, and the scoring function is providing the some binding affinity value or strength of the binding, or we can say a score. So there are several scoring functions. So we don't have time, but I can list here. So like force field based scoring function, empirical methods based scoring function, and knowledge based scoring function. So these are the related software which are based on that, like force fields, it is the molecular mechanics things. Empirical, this is try to reproduce the experimental binding affinities, while these knowledge based scoring functions try to reproduce the experimental geometries, and each method has got their own some of the examples. So, but no software, no scoring function is the perfect. Each has got its own advantage, disadvantage. So it is, if you have the facility, or if you can get, I think the multiple academic licenses are available for multiple softwares, it's better to do docking with multiple softwares. And if we can compare the scoring with the multiple methods, we can have one more category of the scoring functions. We call it consensus docking. So in that way, we can trust the results. If suppose you are using 3 docking softwares, and out of the at least 2 are giving similar trends in your docking experiment, then you can trust the results. Otherwise, it may be possible that one software giving the good results, another giving the negative results, then what to trust. So it is better to, if we can get the more softwares. Yeah, so with this we can end. Yeah, one point I will also mention here. So target fishing, you may be knowing, I don't know. So target fishing or reverse docking is also nowadays quite common. So what we do in the docking, so docking, we have one protein and we can have the multiple ligands. So we are trying to see what are the best hits, how they are binding, what is the pose, and what is the possible binding affinity of those ligands in the active site. That's the routine docking. But if you have a ligand and you know that has got some activities, but we don't know where it is, how that ligand is able to produce some action. So in that case, we have the ligand, we have to take multiple proteins and we have to dock across those different proteins. Then we can tell, okay, for that particular ligand, these may be the probable targets. Therefore, it is able to produce multiple pharmacological actions. So this way we are identifying the target. That's why we call it target fishing or reverse docking. So like curcumin is a quite famous example. It has got a multiple pharmacological activity, and you can determine how this molecule is able to produce their action by exploring the targets in inflammation, or in this area, antioxidant area, memory related targets. So with this, I thank you for listening me patiently, and I'm sorry for there was some internet issue was there. So if you have any questions, I can try to answer.
Thank you, sir. The lecture was very informative, and it is, sir, hats off to your patience and energy, sir. Thank you, sir.
Yeah, thank you.
Dear participants, if you would like to interact with sir, you can please unmute yourself and you can ask directly.
Good afternoon, sir.
Yeah, good afternoon.
Sir, what is the tool to identify structural water, sir?
Yeah, PyWater is there that you can use. That's freely available tool, and I have not used it, but nowadays several, if you Google it, you will get several options to check conserved water molecule. But PyWater, I know that's one of my friend has developed during his PhD.
Thank you, sir. And one more question, sir.
Yeah, please.
Sir, to validate the docking scores, docking validation, If we have done the homology modeling of the protein, how to validate that protein docking, sir? I mean,
Yeah. So it is challenging that if we do not have the protein bound any ligand, and if we are developing homology model, of course we won't have. So you have to see, means when you are going to develop a homology model, so you are going to take some templates. So you see, if by chance if your template has got already some ligand, then you can copy and minimize the structure. You will get some tentative idea how that ligand can bind. Otherwise you have to go for literature. Means some already inhibitors may be reported in the literature. Okay, this is the most potent inhibitor and this is the less potent or inactive molecule. Then you can compare that your most active molecules should have the better score compared to the inactive or less active molecule. I think that's the one way you can try to validate your method for your target.
thank you so much, sir.
Yeah, Ambuja, you can ask your query.
Sir, good afternoon.
Yeah, good afternoon.
Sir, first of all, thank you for very informative talk. The question is, you were talking about the united approach. The thing is, sir, in silo it's okay, but what about if like we are choosing the sites from different conformation, but actually in reality, does it allows like proper folding conformation?
So why you are worrying about folding conformations? Just
Sir,
you have to worry about only your active sites. So if you having facing in generation of a unified model, you can go for ensemble based docking where you will have the more options, more feasibility, more conformers.
yeah, but in actually the binding side, what we learn, it is not just the planar, but actually it is a three-dimensional structure. And if we choose like this, I mean, whether in vivo it allows, that is the question, sir.
Yeah, so unified you will use, okay, you have the three structure or two structure, three structure, right? But in the case of ensemble docking, you can use the limitless conformers, right? You can use many conformers. You can use like you perform MD of 100 nanosecond, you choose the 100 conformers. And out of 100 conformers, if you are able to get your result in 70%, 60%, it's good enough, right? So that will explore more chemical space or conformation space of your target. But if we have only three, means just it's very three only, right? Yeah, if it is coming from NMR, then you have more chances that you can try to combine, because where we do not have lattice forces.
Okay, thank you, sir.
Sir, can you stop sharing the screen, sir?
Yeah, please.
Participants, if you want to interact, you can. Otherwise, we'll go for well, thanks. If anyone wants to interact, please. Sir, from Department of Chemistry and Forensic Science, we would like to present you a certificate of appreciation. So kindly accept, sir.
Yeah, thank you very much, ma'am. Thank you.
Thank you, sir. Now I invite Ms. Anjum Fatima, Assistant Professor in Department of Chemistry, to present vote of thanks to sir.
Thank you, ma'am. Am I audible, ma'am?
Yes, you are audible.
Good evening, everyone. It's my privilege to have been asked to propose vote of thanks for today's session on molecular docking and its application in structure-based drug discovery. I, Anjum Fatima, on behalf of the chemistry department, extend a very hearty vote of thanks to Dr. Devendra Kumar Dakar for giving an excellent coverage on the topic. Sir has enlightened us the use of computer-aided drug designing, molecular docking, protein ligand binding, binding mode and binding site, unique properties of binding sites, various molecular interactions, binding of metalloligand or metallodrug binding with proteins, ligand protein docking, docking validation, visual inspection, so on. So the way sir explained the topic is exemplary. Thank you so much, sir, for the fantabulous presentation. I also wish to express my gratitude to our principal, Dr. J. Achita Devi ma'am, and also our beloved HOD, Dr. V. Jivanjyoti ma'am. And finally, a heartfelt thanks to all the participants. Thank you. Have a nice day.
Yeah, thank you.
Thank you, sir. Dear participants, please fill the feedback form.